Number 10. Cheddar Man. The face Britain never expected in the limestone cliffs of Somerset, England. Gough's cave opens into a darkness that once sheltered people at the edge of a changing world. In 1903, excavators inside that cave uncovered the incomplete skeleton of a man who had died nearly 10,000 years ago. He became known as Cheddar Man, one of the oldest human remains found in Britain. For decades, his bones proved that people had lived on this island long before farms, towns, monuments, or anything later generations would call Britain. Then scientists recovered ancient DNA from his remains, and the skeleton became something more unsettling, a challenge to the face people had imagined for their own distant past. Cheddar Man lived during the Mesolithic, after the worst cold of the Ice Age had retreated, but before farming arrived in Britain. Around him lay forests, rivers, caves, and shifting coastlines, traveled by small communities who hunted and gathered in a landscape whose names are lost forever. Genetically, he belonged to the Western hunter-gatherer ancestry found among prehistoric populations across parts of Europe, before later migrations brought new people and new genetic traits from the southeast, and, much later, from the steppe. His world was not the beginning of a stable national bloodline. It was one buried layer in an island that would be rewritten again and again. The discovery that seized public attention came when researchers studied genetic markers associated with appearance and helped construct a possible face. In 2018, the Natural History Museum in London presented Cheddar Man with dark to dark brown skin, dark hair, and blue or blue-green eyes. The image was striking because it violated a modern assumption that the earliest inhabitants of Northern Europe must already have looked like many Northern Europeans today. A man from prehistoric Britain was suddenly staring out from the museum with a face many viewers had never expected, and a scientific reconstruction became a cultural shock. Yet DNA does not preserve a photograph. It provides evidence from which researchers estimate probabilities. Skin pigmentation depends on many genetic variants acting together, and ancient DNA can be damaged or incomplete. The reconstruction did not mean that science had recovered an exact shade of skin or reproduced every feature Cheddar Man possessed. The evidence supported a narrower conclusion. He was unlikely to have belonged to the lightest pigmentation categories common in much of Britain today, while his markers were consistent with dark hair and light-colored eyes. Similar patterns in other prehistoric European hunter-gatherers suggest that he was not an impossible exception, but part of a Europe whose appearance changed over time. That change was written through migration. The people who shared Cheddar Man's ancestry did not remain alone on the island. Thousands of years later, farming communities connected to Anatolia reached Britain and transformed its way of life and much of its genetic composition. Later still, groups associated with the Eurasian steppe reshaped the island again. Traits now imagined as ancient or permanent were not fixed from the beginning. They became common as newcomers absorbed or replaced earlier communities. Cheddar Man's genome does not offer the comforting image of an unchanged people rooted in one land. It exposes a Britain built from survival, disappearance, and repeated arrival. This is why his face became controversial. To some, the reconstruction corrected misleading images projected onto prehistoric Europe. To others, it became a political threat, as though the appearance of one Mesolithic hunter-gatherer could decide who belonged in a modern country. Both reactions reveal the danger of forcing an ancient individual into identities that did not yet exist. Cheddar Man was not a symbol walking through the forests of Somerset. He was a human being from a population that later lost much of its distinct genetic presence, a man whose remains endured while the world that gave him meaning vanished around him. Almost everything he knew disappeared, long before his skeleton was found. No genome can restore the language he heard, the people he loved, or the name his community gave the cave in which his bones endured. Ancient DNA recovered only fragments, clues to ancestry, clues to appearance, and evidence that the past is often stranger than the images modern people inherit. The controversy was never truly about whether one face could define Britain. It was about the shock of realizing that it could not. Beneath the island's familiar history lay older people, older appearances, and lost communities that a damaged genome forced back into view. Cheddar Man did not reveal an unknown species. He revealed something quieter and more unsettling. Even when an ancestor's bones survive, the past may still wear a face the future refuses to recognize. Number 9. Lake Mungo Man. The ancient DNA discovery that collapsed in the dry basin of the Willandra Lakes region in southeastern Australia. The wind moves across a landscape that once held water, life, and human memory. There, in 1974, the remains known as Lake Mungo III, or Mungo Man, were uncovered from ancient sands. Buried more than 40,000 years ago, he became one of the oldest known human burials in Australia, and a figure of profound importance to Aboriginal history. His presence already carried an extraordinary message. People had lived, mourned, and practiced ritual on this continent at a depth of time almost impossible to imagine. But in 2001, Mungo Man became the center of acclaim, one that seemed ready to fracture the accepted story of modern human origins. A research team announced that it had recovered mitochondrial DNA from his remains. This form of DNA traces only a maternal line, yet the sequence attributed to Mungo Man seemed unlike the maternal lineages found among living humans. It appeared to fall outside modern Aboriginal Australian diversity and outside the broader variation of living people altogether. If genuine, it suggested that a deeply divergent human lineage had survived in Australia tens of thousands of years ago. For those searching for a lost branch of humanity, the ancient lakebed appeared to have delivered genetic evidence of people whose ancestry had otherwise vanished. The danger lay in how fragile that evidence was. Early ancient DNA research often relied on polymerase chain reaction, or PCR, to copy tiny fragments of genetic material until there was enough to study. PCR was powerful, but it could not know whether a molecule came from an ancient skeleton or from a modern person who had touched the sample after excavation. In a human bone being tested for human DNA, contamination could hide almost perfectly. A few modern cells introduced during handling, museum storage, examination, or laboratory work could become louder than the genuine ancient signal researchers hoped to recover. Australia was already one of the most difficult environments in which to preserve ancient DNA. Heat drives genetic material to break apart until authentic sequences may disappear. Mungo Man's remains had also passed through decades of handling, creating repeated opportunities for foreign DNA to enter the sample. Still, the original result spread widely because of what it seemed to mean. The burial became a possible challenge to the foundations of human evolution. An apparent maternal lineage so old and isolated that it seemed to exist outside the known family of living humanity. Then the discovery was tested again with methods the earlier era did not possess. In 2016, researchers reanalyzed Lake Mungo material using improved sequencing and DNA capture approaches built to separate ancient molecules from modern contamination. Genuine ancient DNA should not look fresh. It is expected to be short, broken, and marked by characteristic chemical damage accumulated after death. These patterns help researchers judge whether a sequence truly survived from deep time or entered a sample recently. The new analysis did not confirm the lost lineage. It found no identifiable Aboriginal Australian mitochondrial DNA in the Mungo Man sample. Instead, it detected multiple modern European mitochondrial sequences, genetic traces introduced long after his death. The anomaly that once seemed capable of rewriting human prehistory was no longer evidence of an unknown population. It was evidence that modern contamination had been amplified and mistaken for a message from the ancient past. The reversal made Lake Mungo one of the most important cautionary cases in paleogenomics. It did not diminish Mungo Man himself. His burial remained ancient, extraordinary, and meaningful. What collapsed was the claim that his DNA revealed a secret evolutionary branch. Cases like this forced the field to become more demanding, requiring clean laboratories, protective handling, contamination estimates, independent confirmation, and damage patterns expected from genuinely ancient molecules. There is a darker reason the case still matters. Ill matters. Extraordinary genetic claims gain power because they appear to recover histories that have been ignored or denied. Mungo Man already belonged to a human past of immense depth, connected to living Aboriginal communities, and to a continent whose ancient story did not require a sensational anomaly to be profound. Yet for a time, the promise of an impossible lineage threatened to overshadow history preserved in his burial. Lake Mungo did not reveal a ghost population hidden beyond the human family tree. It revealed how easily the dead can be made to speak with the genetic voice of the living. In a field where a few molecules can redirect the history of entire populations, the most dangerous discovery is sometimes the one that arrives before anyone has proved it truly came from the past. Number 8. Ancient Egyptian mummies. The 3 genomes that ignited an identity war few civilizations have been claimed, reimagined, and fought over as fiercely as ancient Egypt. Its kings, temples, tombs, and painted faces have become symbols far beyond the Nile Valley, pulled into modern arguments about Africa, the Mediterranean, the Near East, race, nationality, and cultural ownership. Yet beneath those competing images lay a scientific obstacle. Egypt preserved bodies in extraordinary numbers, but its climate was hostile to evidence that could speak directly about ancestry. Heat, humidity, embalming chemicals, and centuries of handling all worked against the survival of ancient DNA. Then, in 2017, researchers announced that they had recovered genetic data from mummified individuals buried at Abu Zair el-Malek, an archaeological site in Middle Egypt. The cemetery covered a vast stretch of time, from periods connected to the later New Kingdom through the Greek and Roman eras. The technical achievement was real. For generations, Egyptian mummies had been viewed as nearly hopeless candidates for reliable genetic recovery, their preserved appearance concealing how badly their DNA might have decayed or been contaminated. By extracting material from teeth and dense bone, where fragments can survive better protected than on exposed surfaces, the researchers recovered 90 mitochondrial genomes and genome-wide nuclear data from 3 individuals. That distinction is the key to everything that followed. Mitochondrial DNA traces only one narrow maternal line. It can reveal important patterns, but it does not represent the full ancestry of a person, much less an entire civilization. Nuclear DNA reaches across the wider genome, but here, the complete genome-wide evidence came from only 3 people, all buried at the same site. The findings suggested that the individuals from Abu Zair el-Malek had strong genetic affinities with ancient populations of the Near East, and that modern Egyptians in the comparison carried more ancestry related to sub-Saharan African populations than the ancient samples examined. In the study's models, that later increase was estimated at roughly 8%. Scientifically, this was a careful result from a specific cemetery. Publicly, it became something far larger. Headlines and online debates treated a narrow sample as though it had answered the identity of every person who ever lived under the pharaohs. Some seized on the Near Eastern affinity to argue that ancient Egyptians were detached from Africa. Others rejected the study as an attempt to remove Egypt from African history. Yet both readings demanded more from the evidence than it could provide. Genetic closeness to ancient Near Eastern groups does not mean that the sampled people were not Egyptian, and it certainly does not transform them into a modern European population. Egypt stood at a crossroads of Africa and Western Asia for thousands of years, with trade, empire, marriage, migration, captivity, and conquest leaving layers no single cemetery could contain. What the study did not recover may be as important as what it did. Abu Zair el-Malek lies in Middle Egypt. It cannot automatically represent southern Egypt, Nubia, the Delta, desert oases, frontier settlements, royal households, workers' villages, or people living outside the sample range. A burial ground may preserve a local community with its own ancestry patterns, or a group shaped by status and geography. 90 maternal lines reveal depth within one location, but 3 genome-wide individuals cannot carry the genetic burden of a civilization that endured for millennia along an entire river valley. The controversy exposed a recurring danger in ancient DNA. The moment a result touches identity, precision begins to disappear. Words such as affinity, admixture, and sampled population are technical and limited, but they are easily replaced by absolute categories the ancient world did not recognize. The people buried at Abu Zair el-Malek could not have known that fragments of their remains would one day be used in battles over modern belonging. Their DNA was not a verdict on who owns Egypt. It was a partial record of one community inside a vast and changing human landscape. That is what makes this case unsettling. The breakthrough was genuine, yet the mystery grew larger because the evidence was asked to answer a question it was never large enough to settle. Ancient Egypt did not emerge from 3 genomes, and it will not be contained by them. Somewhere beyond that single cemetery, in untested tombs and unpreserved bodies, stretching from the Mediterranean to Nubia, the fuller genetic history of Egypt remains buried, fragmented, and silent. Until more of those voices are recovered, the most controversial thing about the mummy DNA is not what it proved, but how much the living wanted it to prove for them. Number 7. Haplogroup X. The Native American lineage hijacked by a lost Europe theory. Some mysteries become controversial, not because the evidence is false, but because a genuine gap is pulled toward the wrong conclusion. In the maternal ancestry of indigenous peoples of the Americas, one such gap is known as haplogroup X. Mitochondrial DNA passes through the maternal line, preserving only one narrow branch across generations. Most Native American maternal lineages are described through haplogroups A, B, C, and D, with connections to ancient populations of Northeast Asia and Beringia. Then there is X, rarer, more elusive, and easier to misunderstand. The Native American branch is called X2A. It appears in a minority of indigenous populations and belongs to the larger haplogroup X family, also found across portions of Eurasia, including Europe. That broad distribution created an irresistible temptation. If a maternal lineage appeared on both sides of the Atlantic, some asked, could it be evidence that Ice Age Europeans reached North America before the ancestors of later indigenous peoples? Could it be the genetic fingerprint of a forgotten ocean crossing? That question became entangled with the Solutrean hypothesis, a proposal that people associated with the Solutrean culture of Ice Age southwestern Europe crossed the Atlantic and helped create the Clovis tradition in North America. Supporters pointed to supposed similarities between European and Clovis stone tools. Haplogroup X appeared to offer something stronger than a resemblance in stone, a biological connection. In popular retellings, a rare maternal lineage became the shadow of lost Europeans walking through prehistoric America, but genetics did not say what the theory needed it to say. Native American X2A is not a direct match for known European lineages. It is its own distinct branch, separated by deep time from the forms of haplogroup X found in Europe and Western Asia. If a European founding population had carried this lineage into the Americas, researchers would expect broader genome, wide signs of that ancestry, not merely one isolated maternal branch. Those signs have not appeared. The mystery is real, but it lies somewhere else. The missing piece is Beringia, the vast Ice Age landscape that once linked Siberia and Alaska when sea levels were far lower than today. The ancestors of Native American populations passed through or remained within that region before dispersing into the Americas, carrying only part of the genetic diversity of earlier Asian populations. In small founding groups, rare lineages can survive by chance while disappearing from the populations they left behind. Later, migration and genetic turnover in Siberia could erase the relatives of X2A from the modern record. A maternal branch may endure in the Americas even after its closest source lineages vanish from the land where it began. This is the difference between a missing ancestor and a European one. The absence of an obvious X2A relative in modern Siberian populations does not open a secret route from Europe across the Atlantic. It reveals how incomplete the genetic record can become after thousands of years of movement and replacement. The lost lineage may once have belonged to a Beringian population, whose remains have not survived, whose descendants were absorbed, or whose burial grounds now lie beneath transformed landscapes. Ancient DNA made the distinction sharper. Pre-contact individuals from the Americas carried X2A, related ancestry long before European colonization, proving that the lineage was ancient on the continent, not recently introduced. More decisive still, genome-wide data from early Native American remains, including the Clovis-associated Anzic child, connected those populations to indigenous ancestry and Northeast Asian or Beringian origins, not to a special European founding group. The stones could be debated. The larger genetic record was far less forgiving. Haplogroup X remains haunting precisely because it preserves an absence. Somewhere in the ancient population history of the North, a maternal lineage entered the Americas and then lost the visible trail leading back to its source. That disappearance is mysterious enough, without turning it into a crossing the evidence does not support. The controversy grew because a real blank space in the genetic map was treated as permission to draw an imaginary road through it. What survives is not proof of a lost European America, but a darker truth. Human migrations are filled with lineages that vanish from one region while surviving in another, with entire maternal histories reduced to fragments in living people and ancient bones. Haplogroup X does not show that the first Americans came from across the Atlantic. It shows that even when ancestry survives inside the body, the population that once carried it can disappear so completely that others begin inventing a different origin in its place. Number 6. Anzic. 1. The child whose DNA destroyed an old theory in the foothills of western Montana. A burial waited for more than 12,000 years. The remains belonged to a young child, placed in the earth with stone and bone artifacts stained in red ochre. Among them were tools connected to the Clovis tradition. The fluted projectile points once treated as the signature of some of the earliest widespread people in the Americas. The child became known as Anzic. 1. And although his life ended before he could leave any personal history behind, his DNA would eventually confront a persistent theory about who first reached the American continents. For decades, the origins of Clovis had been a battleground. Their distinctive stone points appeared across ancient North America, and some researchers believed they resembled tools made by the Solutrean culture of Ice Age southwestern Europe. From that resemblance grew an extraordinary proposal. Perhaps European hunters had crossed the Atlantic edge thousands of years before Columbus, reaching North America and helping to establish the Clovis tradition. In this telling, Clovis culture carried a lost European inheritance beneath later indigenous history. The hypothesis found additional fuel in haplogroup X, a rare maternal lineage present in some Native American populations and belonging to a broader family found in Eurasia. Yet tools can resemble one another for reasons unrelated to ancestry, and mitochondrial DNA follows only one maternal thread. What the argument had never possessed was a genome from a human directly associated with Clovis artifacts. Anzic. 1 offered exactly that, not a resemblance in stone or a missing lineage turned into speculation, but the genetic ancestry of a child buried inside the world the theory claimed to explain. In 2014, researchers published the genome of Anzic. 1, and the result was devastating for the idea of a European Clovis founding population. The child did not carry the special European connection the Solutrean hypothesis required. His genome showed close relationship to indigenous peoples of the Americas, with particularly strong affinity to populations in Central and South America, while his deeper origins fit a history connected to Northeast Asia and Beringia. A burial beside Clovis tools pointed not across the Atlantic toward Ice Age Europe, but into the ancestral history of Native American peoples. The finding exposed the limits of archaeology when objects are forced to carry identities. Stone tools do not possess bloodlines. Similar challenges can lead distant peoples to shape stone in similar ways, and techniques can spread without populations being replaced. For years, the claimed resemblance between Solutrean and Clovis technology kept an ocean-crossing story alive. The Anzic genome did not need to debate the angle of a flake or the method used to thin a blade. It revealed who one Clovis-associated child belonged to, and that answer contradicted the theory at its center. There was another consequence that could not be separated from the science. Anzic. 1 was not simply a specimen from a vanished population. His genome revealed relationship to living indigenous communities, transforming archaeological material into the remains of an ancestor. The research involved consultation with Native American groups, and the child was eventually reburied. Ancient DNA can recover hidden history, but it can also repeat old harm when the dead are treated as data alone, rather than as people whose descendants are still here. What disappeared at Anzic was not the ancestry of the child, because that survived across the Americas. What disappeared was the recognizable world of Clovis. The communities who made those tools spoke languages now lost, buried their dead, and later transformed into traditions archaeology can trace only imperfectly. When artifacts remained but voices were gone, others could attach new origins to them. A missing cultural name became an opening into which a lost European founding story could be placed. The genome did not solve every question about the first peoples of the Americas. Earlier migrations, coastal routes, population splits, and Ice Age shorelines drowned beneath rising seas still conceal evidence. But on one critical point, Anzic 1 changed the argument forever. The Clovis child was not proof of an erased European beginning. He was evidence of deep indigenous continuity, a direct link to peoples whose ancestors endured across the continent. A child who never lived long enough to speak left behind an answer stronger than decades of speculation. His tools could be compared and debated, but his genome could not be made to carry a history it did not contain. Beneath the red ochre of a Montana grave, ancient DNA did not reveal a forgotten crossing from Europe. It returned one of North America's oldest known children to the ancestry that had been his all along. Number 5. Polynesia and the Americas. The ocean-crossing DNA remembered the Pacific Ocean is so vast that it once seemed capable of separating human worlds forever. Across its open waters, the islands of eastern Polynesia rise like isolated fragments of land, thousands of miles from the coast of South America. To many later observers, those distances looked impossible for ancient contact. Yet Polynesian navigators did not see the sea as an empty barrier. They crossed it by stars, swells, winds, birds, and generations of knowledge carried in voyaging canoes. Long before genetics entered the question, one clue suggested that this ocean may have concealed an encounter history had forgotten. The sweet potato, a crop native to the Americas, was already growing across Polynesia before European voyages connected the regions on written maps. For decades, that clue remained suspended between possibility and doubt. Plants can travel in unexpected ways, words can resemble one another by coincidence, and oral histories are often dismissed when they do not fit the maps drawn by outsiders. Rapa Nui, the island later called Easter Island, stood near the far eastern edge of the Polynesian world, facing an ocean that stretched toward the American coast. If voyagers from these distant shores had once met, their canoes had decayed, their landing places had eroded, and the scale of contact may have been too small to leave monuments or battlefields behind. The Pacific kept it secret because almost everything used to cross it was made to vanish. Then researchers turned to the evidence carried inside living descendants. In 2020, a genome-wide study examined DNA from Polynesian populations across the Pacific and found segments of Native American-related ancestry in several groups. This was not the vague echo of a legend or an argument based only on a transported crop. The inherited fragments could be modeled through time, and their estimated arrival pointed to approximately 1200 CE in parts of eastern Polynesia, centuries before sustained European contact. Some comparisons suggested affinity with indigenous populations connected to the northern regions of South America. Suddenly, the ocean no longer looked silent. It carried the genetic remains of a meeting that no European chronicler had witnessed. A finding based on modern people could always face one challenge. Perhaps the ancestry entered later, during the chaotic centuries after European expansion transformed the Pacific. Movement, enslavement, disease, colonial ships, and new settlements created countless opportunities for recent mixing. A contact event dating before Columbus required stronger evidence, ideally from people who had lived closer to the period before those later disruptions. That evidence arrived through ancient genomes recovered from Rapa Nui itself. In 2024, scientists reported DNA from 15 ancient Rapa Nui individuals, dating from the 17th century into the 20th. Their genomes carried Native American ancestry as well, at around 10%, with modeling placing the original contact between roughly 1250 and 1430 CE. The signal had survived in bodies far less vulnerable to explanations based on very recent mixing. What the DNA proved was contact, but it did not restore the voyage itself. No genome can show the shape of the canoe on the horizon, the first exchange on a beach, the language spoken between strangers, or the direction in which the journey began. Polynesian navigators were capable of extraordinary long-distance voyages, making travel eastward toward the Americas entirely plausible. It is also possible that Native American voyagers moved westward into the Pacific, or that movement occurred in more than one direction. The genetic evidence preserves the meeting, but the ocean has kept the faces of those who first crossed it. This is what makes the case so powerful. For generations, the Pacific was described as a boundary so enormous that human contact before European exploration could be treated as fantasy. Yet the people of Polynesia inhabited a navigated world, not an empty wilderness of water. Their expansion across the ocean remains one of humanity's greatest achievements, and DNA now reveals that somewhere at its far edge, their history touched the history of the Americas. The sweet potato was not standing alone anymore. It had been joined by ancestry. The lost world here was not a vanished species or a buried kingdom. It was a moment between peoples, a meeting on an ocean route, whose records were carried in bodies rather than books. The canoes disappeared. The shores changed. Colonial history obscured what earlier voyagers may have known, but fragments of that crossing endured from one generation to the next, silent until science learned how to read them. The Pacific did not keep two human worlds apart. It hid the evidence that they had already found one another, long before history was prepared to believe it. Number 4. The Neanderthal genome, the extinct humans still living inside us for most of modern archaeology. Neanderthals occupied a lonely place in the story of humanity. Their heavy-browed skulls, powerful bodies, stone tools, and Ice Age caves suggested a people close enough to resemble us, yet separate enough to be placed on a dead branch of evolution. They had lived across Europe and Western Asia for hundreds of thousands of years, endured climates that repeatedly transformed the continent, and then disappeared around 40,000 years ago as modern humans spread through their world. The familiar image was clean and final. One kind of human arrived, another faded away, and the Neanderthals left behind bones, tools, and no living descendants. That image survived because fossils could only reveal so much. A skull can show anatomy, and artifacts can show behavior, but neither can prove what happened when two populations encountered each other. For decades, researchers argued over whether modern humans replaced Neanderthals or mixed with them. The answer was locked inside ancient bones, but retrieving it seemed almost impossible. After death, DNA fragments into tiny damaged pieces, while microbes flood the remains with their own genetic material. Worse, any modern person handling a Neanderthal bone can leave behind human DNA similar enough to confuse the signal. In 2010, a team led by Svante Pääbo published a draft Neanderthal genome reconstructed from ancient remains. Instead of relying on the appearance of bones, researchers could compare extinct human DNA directly with genomes from living people. The result shattered the old picture. Neanderthals shared more genetic variants with present-day people outside sub-Saharan Africa than with people whose ancestry remained primarily within Africa. The strongest explanation was interbreeding. After some modern humans left Africa and entered Eurasia, they encountered Neanderthals, had children with them, and carried fragments of Neanderthal ancestry forward as their descendants spread across the world. The original estimates suggested that living non-African populations carried roughly 1 to 4% Neanderthal ancestry. That number can sound small until its meaning is understood. Neanderthals had not disappeared in the absolute sense. Their communities vanished, their languages fell silent, and no one continued to call themselves Neanderthal, but portions of their genome survived inside billions of human beings. Extinction no longer meant a lineage had been erased completely. It could mean that a people ceased to exist as a distinct population while pieces of them remained inside those who came after. The discovery also revealed that survival was selective. Modern genomes do not preserve Neanderthal DNA evenly. Some inherited segments persisted because they were neutral or may have offered advantages in unfamiliar Eurasian environments, including influences connected to immunity, skin biology, and metabolism. Other regions show a striking absence of Neanderthal ancestry, suggesting that natural selection removed combinations that created biological costs in modern human descendants. The encounter was not simply the merging of two peoples. It was a long aftermath in which some inherited fragments remained and others were slowly purged. The controversy was immense, because the genome overturned a boundary that generations of diagrams had made appear permanent. Neanderthals were no longer only evolutionary cousins whose story ended before ours fully began. They were part of the ancestry of much of the living world. The old tree of humanity, with clean branches separating and never touching again, gave way to something darker and more complicated: populations splitting, meeting again, exchanging genes, then vanishing as recognizable peoples, while their inheritance traveled onward under different names. There is a sadness hidden inside this discovery. A surviving genetic fragment cannot tell us what a Neanderthal mother called her child, what memories passed between communities, or whether those encounters came through alliance, desperation, violence, or affection. DNA confirms that contact happened, but the lives behind that contact remain beyond recovery. Their bones emerge from caves as isolated remains. Their inherited genes survive without the stories that made them human. The Neanderthal genome did not resurrect an extinct people. It revealed that extinction had never been as complete as we imagined. Somewhere within the immune responses, bodies, and inherited variation of living humans, a population once believed to have vanished without descendants still leaves its mark. The most unsettling part is not that Neanderthals were different from us. It is that after tens of thousands of years of disappearance, the line separating them from us was never clean at all. Number 3. The Denisovan gene that let Tibetans survive the roof of the world high above the valleys of Asia. The Tibetan plateau rises into a world where ordinary human survival begins to fail. Much of this landscape stands more than 4,000 meters above sea level, a place of frozen grasslands, hard winds, and air so thin that every breath carries less oxygen than the body expects. For newcomers, the consequences include exhaustion, dangerous strain on the body, and risks during pregnancy. Yet for countless generations, Tibetan communities have lived, worked, and raised children at this altitude, as though their bodies carried an answer to a problem the mountain gives to everyone else. That answer was eventually traced to a gene called EPase-1, part of the system that helps the body respond to low oxygen. Most people arriving from lower elevations react by producing more hemoglobin, the molecule in blood that carries oxygen. At first, this seems useful, but too much hemoglobin makes the blood thicker and places greater strain on the body. Tibetans show a different pattern. They cope with chronic oxygen shortage without depending on the same dangerous rise in hemoglobin. Researchers searched for the inherited change that made permanent life at altitude possible. When scientists studied the Tibetan version of EPase-1, they found something deeply unusual. Its genetic pattern did not look like an ordinary variant that had slowly emerged within modern human populations. The haplotype, a linked stretch of inherited DNA, was far more divergent than expected and strikingly common among Tibetans, while remaining rarer in nearby Han Chinese populations. This was not simply a random difference between neighbors. Natural selection had favored it in the one environment where oxygen scarcity could decide who endured and who could safely bring children into the world. Then researchers compared that strange genetic signature to the genome of a human population almost absent from the visible record, the Denisovans. Denisovans had been revealed through fragments recovered from Denisova cave in Siberia, initially represented by little more than a finger bone and several teeth. Unlike Neanderthals, they left no familiar library of skeletons, yet the Tibetan EPase-1 variant matched Denisovan, like DNA far better than ordinary modern human variation. A population known through a handful of ancient remains had left a genetic inheritance powerful enough to shape life across one of the harshest inhabited landscapes on Earth. The strongest explanation is adaptive introgression. Long ago, ancestors of some modern humans encountered Denisovans, had children with them, and carried away fragments of their DNA. Much of that inherited material may have faded or offered no lasting advantage, but when people began living at high elevations, the Denisovan, like EPase-1 variant, became extraordinarily valuable. Those who carried it were better equipped for the thin air, and over generations the gene became common. Not because Denisovans survived on the plateau as a separate people, but because one piece of their biology did. This discovery changed the meaning of archaic ancestry. DNA inherited from extinct humans was often imagined as a residue of ancient encounters, something modern evolution would eventually remove. EPase-1 revealed the opposite possibility. A vanished population could pass on a solution modern humans did not yet possess. In Tibet, Denisovan, like DNA, was not merely proof of forgotten interbreeding. It became a survival tool, helping human communities live where the atmosphere itself places limits on the body. The extinct did not simply fade into the past. They helped another lineage inherit a future at extreme altitude. The loss behind this case remains immense. We know almost nothing about Denisovans as people. We do not know what they called themselves, how widely they traveled, how they survived different environments, or how many times they met the ancestors of living populations across Asia and Oceania. Their fossil record is so sparse that without genetics, their existence might have remained almost invisible. Kingdom may leave ruins, and a settled people may leave cemeteries, but the Denisovans left something stranger, a biological advantage hidden inside humans living thousands of years after their own population disappeared. On the Tibetan plateau, Every breath carries the echo of that lost inheritance. The mountains preserved no Denisovan cities, and the caves revealed only fragments of bodies. But inside the physiology of living Tibetans, ancient DNA preserved the consequence of a meeting no written history could remember. A vanished human group, reduced in the archaeological record to scattered bone and teeth, left behind a gene that helped another people survive above the clouds. The Denisovans may have disappeared as a population, but on the roof of the world, part of them never stopped breathing. Number 2. Cima de los Huesos. The Neanderthal bones with Denisovan DNA deep beneath the Sierra de Atapuerca, in northern Spain beyond narrow passages and limestone darkness, lies a vertical shaft known as the Cima de los Huesos, the pit of bones. At its bottom, researchers uncovered one of the most extraordinary human fossil collections from the Middle Pleistocene. The remains of at least 28 individuals had accumulated there. Skulls, jaws, teeth, limb bones, lives sealed away for roughly 430,000 years. Long before DNA could be extracted from them, their anatomy seemed to tell a recognizable story. Their faces and skulls carried early traits associated with the Neanderthal lineage, as though this chamber preserved a population already moving toward the humans, who would later inhabit Ice Age Europe. That conclusion made sense because bones were once the clearest archive available. Brow ridges, facial structure, teeth, and skeletal proportions allowed scientists to place ancient humans along evolutionary paths, even when the details remained uncertain. The Cima individuals appeared to be early Neanderthal relatives, older than the classic Neanderthals recovered from later European sites, but visibly connected to them. Nothing about that interpretation suggested that these bones would one day point toward another extinct population, known mainly from a Siberian cave. Recovering DNA from material this old was almost impossible. At more than 400,000 years, genetic material has usually been shattered beyond recognition, leaving fragments damaged and surrounded by contamination. Scientists first targeted mitochondrial DNA because each cell contains many more copies of it than nuclear DNA, giving the molecule a better chance of surviving. From one Cima individual, researchers reconstructed a nearly complete mitochondrial genome, enough to compare its maternal ancestry with that of other extinct humans. The expected result was simple. Neanderthal-like anatomy should carry Neanderthal-like mitochondrial DNA. Instead, the sequence clustered closer to Denisovans. These were the mysterious archaic humans identified primarily through genetic material recovered in Denisova Cave in Siberia, a lineage nearly invisible in the fossil record. In the darkness of a Spanish cave, a skeleton that seemed to belong on the road toward Neanderthals carried a maternal signal tied more closely to their eastern relatives. The bones pointed one way, the inherited line from the mother pointed another. For a moment, the finding threatened to turn the human family tree into confusion. Had Denisovan, related humans once lived across Europe? Were the Cima fossils not connected to Neanderthals at all, or had an ancient maternal lineage survived in Europe before later disappearing? The picture became clearer only when researchers succeeded in recovering nuclear DNA, the broader genetic record. That signal placed the Cima population firmly on the Neanderthal lineage. Their anatomy had not lied, but neither had the mitochondrial sequence. The same bodies were preserving two histories at once. This contradiction is called mitochondrial nuclear discordance. Mitochondrial DNA follows only a single maternal thread, while nuclear DNA captures the far larger ancestry of a population. A group can be closely related to one lineage overall, while still carrying a maternal branch inherited from older contact or population structure. At Cima de los Huesos, the individuals may have carried an early mitochondrial lineage related to Denisovans, one that was later replaced among later Neanderthals by another branch. What disappeared was not merely a name on a family tree, but a line of mothers whose genetic signature endured for a time and then vanished from later descendants. Without ancient DNA, that disappearance would have remained invisible. Fossil anatomy could reveal that the Cima people belonged close to Neanderthals, but bone shape could never show which maternal lineage they carried or when it was lost. Pit of bones forced researchers to abandon the image of human evolution as a clean tree with branches that separated forever. Ancient humans could divide, meet again, exchange ancestry, and lose entire genetic lines while retaining others. A body could look like the ancestor of one vanished people, while preserving the genetic shadow of another. More than 400,000 years ago, the people in that shaft left no names, voices, or explanation for the strange history hidden inside them. Yet when science finally opened their damaged genetic archive, the cave revealed something more haunting than a new fossil. Inside bones shaped by the Neanderthal story, a maternal shadow was still waiting, pointing eastward toward Denisovans, and proving that even the oldest human identities were never written in a single line. Number 1. The ghost humans hidden inside West African DNA. Every case so far has begun with something tangible: bones, graves, or fragments of extinct humans carried into the present through interbreeding. But the final discovery begins with an absence. No skull has been placed in a museum, no jaw matched to a vanished face, and no named grave. Instead, researchers found the trace of an unknown human population inside the genomes of living people in West Africa, a genetic shadow left behind by ancestors whose physical remains have never been identified. Simplified versions of human origins once treated Africa as a single starting point, the homeland from which modern humans emerged before spreading outward into the world. But Africa is not a blank beginning to the story. It holds the deepest human genetic diversity on Earth, shaped by populations that separated, met again, and exchanged ancestry long before written memory. In the genomes of populations including Yoruba, Isan, Mende, and Gambian groups, scientists detected patterns that did not fit a clean history of one uninterrupted modern human lineage. The evidence came from inherited blocks of DNA far more divergent than expected, as though they had entered the ancestors of living West Africans from a population separated from them for an immense period of time. Researchers modeled these fragments and arrived at a disturbing possibility. Somewhere in Africa, an archaic human lineage may have survived long enough to interbreed with ancestors of modern populations, leaving portions of its genome behind even after the population itself disappeared. Estimates of this hidden contribution vary depending on the population and model used. Because this is not a neatly recovered species, it is the reconstruction of missing people from fragments of inheritance. Scientists call such a lineage a ghost population. The phrase does not mean fantasy, but it also does not mean certainty. It describes a group inferred through genetic effects, even though no confirmed skeleton has yet been assigned to it. Neanderthals and Denisovans eventually became visible through ancient bone and sequenced DNA, allowing direct comparison with living humans. The West African signal is different. There is no ancient genome from the suspected source population waiting to be read, only the pattern it may have left in descendants, like footprints continuing after the person who made them has vanished. The reason physical confirmation is so difficult is part of the tragedy. Much of tropical Africa is brutally hostile to ancient DNA preservation. Heat and humidity break genetic material apart far faster than the cold caves and frozen soils that protected archaic DNA in Eurasia. Bones may survive while the molecular evidence inside them has already dissolved into silence. Entire chapters of human evolution may have unfolded in regions where the Earth destroyed the archive modern science needs. The absence of recovered ancient DNA does not mean the absence of history. It means some environments erase their dead more completely than others. Yet this discovery demands caution, because a ghost can be created by more than one kind of past. The unusual patterns may reflect interbreeding with a distinct archaic population, separated from the ancestors of modern humans for hundreds of thousands of years. But similar signals can also emerge if early Homo sapiens populations within Africa remained isolated for long periods before reconnecting. In that scenario, the ancestry could come from branches of our own species that became deeply divided before mixing again. Both possibilities lead to the same conclusion: the human past within Africa was far more fractured and interconnected than simple diagrams once allowed. What makes the discovery so unsettling is that the unknown population may never receive a face. There may be no preserved tooth yielding a complete genome, and no skull researchers can identify as the missing lineage. The ghost humans of West Africa may survive only in statistical patterns, in segments passed silently through thousands of generations. They may have hunted, migrated, buried their dead, and met other humans whose descendants still live today. Yet everything they were outside those inherited fragments may already be gone. The final mystery of ancient DNA is not confined to what can be recovered from the dead. It is what the living may carry without knowing. Inside modern genomes are remnants of meetings never recorded, peoples never named, and lineages that vanished before history could remember them. The unknown ancestry in West Africa reveals a closed door behind which may lie an entire human world erased from fossils and memory, but not completely erased from us. The most haunting lost bloodline is not the one preserved in a grave, but the one still alive inside people, waiting for science to discover that it was ever there.